All right, this is the fifth and last chapter of the nervous system. So remember that special senses are part of the peripheral nervous system. If you go back to the chart that we looked at when we first started covering the nervous system, special senses falls as that, feeding into the central nervous system, how you perceive your world. This video is going to be, generally speaking, about special senses and then specifically about vision. And then I'm going to have separate videos for taste and smell and hearing. So, senses. There are general senses, specifically of touch, temperature, pressure, pain. We've covered all of those when we talked about the peripheral nervous system, and then we said we would cover special senses later, and now is later. So, special senses are smell, taste. Those both involve chemoreceptors, if you remember, and are we'll cover them together because they are perceived together quite a bit. Sight that we're going to cover now, and then hearing and equilibrium that we are also going to cover together because they both occur in the ear. So we're going to start with vision. For most people, vision is what they think of as their most critical special sense, actually probably their most critical sensory organ altogether, which is interesting in that your eyes are a very small part of your body, and you only see with the very back part of your eye. We'll look at that later. But 70% of all of your sensory receptors are in your eye, and you only see with that little back part. So really that ratio is quite extreme. So that you only, 70% of all of your sensory receptors are in this tiny part of your body. So this is sort of what accounts for the idea that vision is one of our greatest special senses. It does bear understanding that other organisms do not have a very good sense of sight and navigate the world quite well by the sense of smell. Think of things like sharks that have very poor eyesight but have very good chemoreceptors in their noses. There are nocturnal animals that have poor or limited eyesight but have very good hearing and echolocation. So an echolocation is something that we don't even have as a perception. So just understand that not all organisms function on the same evolutionary paradigm. So each eye individually has over 1 million nerve fibers, and the eye has a bunch of different associated organs that go with it. So each eye is in its own bony orbit, which we covered. Remember that part of it is your zygomatic arch. Part of it is the frontal bone, the sphenoid bone, all enclose the eye in bone. The eye itself, I think you probably think that it is, you think that it is more delicate than it is. An eye is made out of tough connective tissue, so it is in fact much more robust than you think. And in addition to that, eyes, if you would dissect an eyeball, there is a thick layer of fat around the eye. And when people lose weight and go through a starvation period, it turns out that they can metabolize the fat that's around the eye. And that's why people who are starving have sunken eyes. It's because they've metabolized the fat that encloses the eye. So talking about accessory structures of the eyeball first, everybody has eyelids. If you were a different kind of animal, you could have a third eyelid that moves in from the side, which would help protect animals that have a lot of sort of assaults in the face area. So animals that live in a jungle or in a grassland where they could be damaged by different types of plant matter, or that hunt with their faces, with their mouths, where their eyes could be more easily damaged. Humans don't. We protect our faces pretty well, so we have one eyelid. The places where they meet, here at the left and the right of the eye, are the medial and lateral canthus. So this is the medial canthus, and this is the lateral canthus. And they are lined with eyelashes, and eyelashes are there to keep dust particles and insects out of your eyes. You have meibomian glands that are inside the eyelids, and these make an oily secretion that helps lubricate the eyes and helps you see well. These meibomian glands can become clogged, I guess, and they can cause styes, which is something you've heard of, which is sort of like kind of like a pimple on your eye. It blocks up, and then the oil builds up inside the eyelid, and it's very, very painful because your eyes are very sensitive. And what you should do in that case, it's interesting, two of my children have just suffered from styes on their eyes, is that it just needs a lot more washing. So if you get some baby shampoo, like Johnson's & Johnson's baby shampoo, and when you're washing your face, you scrub those eyes in the shower with that baby shampoo, it clears up in a day or two. And if you keep up that routine, it won't happen again. So that is the meibomian glands. You also have some ciliary glands. The same thing can happen. It's just less likely because because they secrete sweat, which is mostly saline rather than the oil. But they help make tears. However, we're going to learn more about the tears in a second. All right. You also have a tissue called conjunctiva. That's the membrane that lines the eyelids. It has a connecting function, so it kind of connects, or it touches the surface of the eye, and it makes mucus that also helps lubricate the eye. So all of those together are sort of components of tears. You have probably, as a child, because most children do, have had conjunctivitis. Conjunctivitis is literally just what it means is that it is a swelling or inflammation of the conjunctiva. And that happens for a number of reasons. It could happen because of a virus. It could happen because of allergies. It could happen because of a bacterial infection or even a fungal infection. And what happens in that case is, and this is how you know that you have conjunctivitis, generally speaking, you wake up in the morning and you can't open your eyes because in the night, that conjunctiva that makes mucus has secreted a great deal of mucus, and it has dried. When mucus dries, it becomes a very hard, and it has stuck your eyelashes together. You need to get a wet washcloth and sort of wipe that off. The thing about conjunctivitis is that many forms of it are incredibly contagious. I know that I have caught it for a very, very brief amount of time. I worked with small children, and those small children gave me conjunctivitis, and I've also caught conjunctivitis from my own children. But it is something that you get eye drops from, and it clears up quite quickly. But it has a bunch of different causes, but the result is always the same, that kind of really nasty, itchy, red eyes. And then so I probably should have led with the fact that most people call conjunctivitis pink eye. And that's because when it happens, it causes a great deal of inflammation, even in the sclera, the white part of the eye, and your eyes become quite pink and itchy and painful. and red, and the whole thing is a whole lot of no fun, and it is associated with a great deal of mucus. This being different than an eye cold, which some kids can get. Let's actually move to the next slide, and I'll talk about it. An eye cold, because this nasal lacrimal duct right here connects the nose and the nasal cavities to the eye. Small children it's oriented like this, like laterally up and down in adults, but in small children, because they don't have sinuses yet, it's kind of horizontal. And for little kids, they can have like mucus from their nose move through the nasal lacrimal duct horizontally into the eye, and you can literally wipe their eyes and pull out like boogers from their nose. That is called an eye cold. It is not associated, you think that it's conjunctivitis, but their eyes are not red. And that conjunctiva isn't swollen. It, generally speaking, doesn't bother the children. It only really happens in like toddlers and like infants when they have a cold. It is super duper gross. My children have had it, and it is gross. So you can pull the boogers like right out of their eyes, which is, wrap your head around that. All right, the lacrimal apparatus is what we call the structures that make tears. They are things like the glands that we just talked about, the eye itself, the nasal lacrimal duct, and that sort of canal that moves. There is also a nasal lacrimal sac in, you can see here, the lacrimal sac, and that holds some excess tears. So sometimes if you have dry eye and you pinch the bridge of your nose, the side of your nose, remember back to bones, that's the lacrimal fossa that sits in the lacrimal fossa. And it holds a little bit of extra tears. You squeeze it, and those tears will come out on your eyes. Some of them will go down to your nose, so they can also make your nose run. But if you have dry eyes, it's a good, and no drops apparent, you can squeeze that, and you get extra tears on your eyes. And your tears also, the tears that your body makes, are part of your primary immune system, meaning that they have antibodies in them, and they also have lysozymes, which is an enzyme that breaks down cell walls. So it prevents you from having bacterial or viral infections of your eyes. Your eyes are called an immune privileged site, meaning that they do not have as much immune system as the rest of your body. And so they don't, you can't fight off an infection as well. So if an infection in your eye is quite serious, it's something that can cause the loss of an eye if it's not treated properly. So it's very important that your body fight off an infection before it happens. So that's part of that primary immune system. All right, so you have a bunch of extrinsic eye muscles in the eyeball lab, the vision lab. The eye muscles will be highlighted, but they attach to the eyeball itself. And then they have some of them have, these oblique muscles have like this pulley system, and they pull and move your eyes in a bunch of directions. They are, of course, controlled by cranial nerves, which you learned about 2 chapters ago. And they produce eye movements around, the world. They can move your eyes in all the directions. All right, other parts. So there are 3 general layers. There is a fibrous outside, which is made of the sclera. The sclera is the white part of your eye. And then there's a clear part in the front called the cornea. There is this red layer here, which is the choroid, that has blood vessels and supplies the retina, which is the inside layer that has the nerves in it, with the blood and oxygen that those neurons need. Because remember that your neurons are very needy of oxygen and glucose. And you can have like a stroke in your eye if some of these blood vessels become blocked. And it can cause tissue death and partial blindness if you have a little eye stroke. All right, so the sclera is the white part of your eye. It does have its own blood vessels in it, and if you stay up too late or party too hard, you will find that out. Or if you're sick for some reason, you can see those blood vessels will become dilated, and it causes your eyes to become what we call bloodshot. But generally speaking, if you're healthy and well, that white part is nice and clear, and that is called the sclera. The cornea is the clear part. It's also made out of connective tissue in the front part of your eye, the anterior portion. The cornea is pretty like it can repair itself. And because it does not have, because of the low cellularity of the cornea, because it is connective tissue, it is the tissue that can be transplanted from one human to another. So you can get a cadaverous, like a dead person's cornea, a corneal transplant, and they don't have to do any tissue matching because it is because there's not enough cells for your body to reject it. And also your eye has that immune system that is less robust than the rest of your body, so it's kind of a perfect storm. And for that reason, corneal transplants are fairly easy to come across, so you don't need any matching. All right, so you'll definitely be responsible for the structure of the eye. You have, again, let's just start on the outside. You have the cornea here. It transitions into the sclera. Inside of the sclera is the choroid. The choroid transitions in the anterior portion of the eye into muscles. So you have the ciliary body, which is a bunch of muscles that move this part here, which is the lens. The lens is going to change shape. When you dissect an eyeball, it's actually the hard part. It looks like a hard little marble in there. But in living tissue, it's actually more like a contact lens. So it's kind of squishy and movable. But still fairly firm. And it is moved by these sort of threads. The ciliary body moves the ciliary zonule, or sometimes called the suspensory ligament, and stretches the lens to focus the light that's coming through. So the light comes in through the cornea. It goes through this area here. This is full of liquid. This is called the anterior segment. That's the place that it is. But it's full of fluid called called aqueous humor. Sorry, I just had a momentary lapse. Aqueous humor. It goes through the pupil, which is the hole in the iris. This part is the iris. We'll talk about that in a second. It goes through the lens, and then it goes through the lens. The lens focuses the light. It goes through this back part, which is called the posterior segment, and it should focus on this place called the fovea centralis. The fovea centralis is where the most rods and cones are, specifically cones. And it's the place where you have your best vision. So it's just this tiny little spot. So that's all those things. Meanwhile, the iris is here. The iris is the colored part of your eye, but it's also a muscle, which is kind of wild. And it has two layers on it. One that is circular and one that is radial, meaning that it moves up and down or towards the outside. And it controls how big and how small the pupil is. Again, that's also controlled by your autonomic nervous system, as you learned in the last chapter. So the iris controls the size of the hole in the middle of the iris, which is the pupil. We'll talk about the chambers later. Let's see, what else have we not covered? So the aqueous humor is going to come out from here. This is actually called the posterior segment, the posterior chamber. And it is going to come out, and then it is going to be drained through the scleral venous sinus, which is this thing, which is also called the canal of Schlemm. I feel like I got most things. The ora serrata is here. It's just this dark area, and it's less functional. You do not need to know the function of that. Nerves come out and they exit the eyeball through the optic nerve. There's also blood vessels that come out in the middle, and that actually causes this little blind spot that we'll talk about when we do lab, and that's called the optic disc. All right, the choroid layer. The choroid layer is, your book calls it a blood-rich nutritive tunic. I will not ask you to regurgitate those terms, but it does mean that it has a lot of vascular tissue, a lot of blood vessels, and it wraps around the eye and provides nutrients for the nerves. It is pigmented, so it is darker. If you dissect a sheep or a cow eyeball, you will find that it is sort of iridescent. It is not iridescent in humans. They have an extra layer that makes it sort of look sort of like the inside of a shell. But in humans, it's kind of just dark. And it keeps light, it absorbs some light, so the light doesn't scatter around the eyeball and affect your vision. That would make your vision blurry. Inside, you have a ciliary body that is made of smooth muscle that helps, again, is going to help focus the lens. And then in the front, you have the iris, which is the thing that makes your eyes colored. So if you have blue eyes, your iris is blue. If you have brown eyes, it's brown, green, you know, all that. And then the hole in the middle of it is the pupil. So then we're talking about here, ciliary body, and the iris here, and the pupil in the middle. All right, and then the most functional part is the retina. The retina has photoreceptors in it, and there are two different kinds. There are rods and cones, and we'll talk about them individually in a second. And they are triggered by light, and it causes them to depolarize and send a message from the photoreceptors out. Let's go to this picture actually. So it triggers the photoreceptors and it sends messages from the nerves down the optic nerve out to the brain, into the visual cortex, which we all understand is in the occipital lobe of the brain. We remember that from the central nervous system. All right, so let's talk about some rods. Rods are generally speaking on the outside of the retina, peripherally. They only see in black and white and gray tones, so this is not your color vision, and they are more light sensitive. So this is the reason that if you are out at dusk, particularly when you go from full light to low light, you end up having this period of time where you can still see things, but it's quite fuzzy. And that's because you've stopped using your cones and are only using the rods. So you would find cones around the outside of the retina here, less than you would find it in the back here where light is going to be focused. So looking at the cells themselves, a rod looks like that. That's why they're called a rod. And a cone looks like this. It's cone-shaped, shocking. And the photosensitive part is the outer segment. And then there's a space, and then there's the nucleus. There are cones now, have 3 different types, and they each of them react to a different color. If you are colorblind, then you have one of them that doesn't work. There is a very a rare kind of color blindness where people don't see any color, but for the most part, if you're colorblind, you are lacking one type of cone and you see light in a skewed way. When we do lab, you'll see that. You see, rather than seeing the full spectrum of light that most people see, you see a limited spectrum of color. Cones are generally speaking most dense at the fovea centralis. We'll go look at that in a second. And there are no photoreceptors at all, no rods, nor are there cones at the optic disc, which is your blind spot. So let's go back and look at that. The fovea centralis is here. This is all cones. So when light comes in, again, the lens, if you have perfect vision, is going to focus your light on the fovea centralis here. And that is going to be, if you focus light best and perfectly at a pinpoint on the fovea centralis, then that is, you are going to have very good vision. If your lens does not do that perfectly and the light is not pinpointed at the fovea centralis, then you will be nearsighted or farsighted depending on how that is. And when we do the vision lab, we'll talk about that more specifically. All right, so let's go and look at that. So here you go. Now you're looking at like a little piece of the retina all blown up. So light is going to come in this way or this way. And there are 3 different kinds of cells in the retina. So this is the choroid. There's this pigmented layer, which is dark, and that keeps light, it absorbs light, it keeps it from scattering around. But these photoreceptors are so sensitive that you don't want light shining like right on them. It's kind of counterintuitive, but they face towards the back of the eyeball. So light comes through, light first of all hits these ganglion cells. Ganglion cells are cells that are going to go all the way back to the optic chiasma. And then you have these bipolar cells. We talked about those a little bit when we talked about neurons, and we said that they were in special senses. And the bipolar cells attach to the photoreceptors, the rods and the cones. And so you have them here, and then the photosensitive part, the outer segment of them, are actually facing back towards the back of the eyeball. So light comes all the way through these layers of cells. It's already gone through the whole eyeball like we just talked about, and it depolarizes the rods and cones, if there is enough of it, back here. And too much light can really damage the retina. So you need to, that's one of the reasons that they're facing backwards. So you don't have light damage, because that is a real thing that can damage the retina. And because they're neurons, if you damage your retina, it's very hard to get that that sense back. It's not like it just comes back. All right, this is a scanning electron micrograph of rods and cones. You can see the rods here, and you can see the cones here. This would be a picture of cells at the periphery, because you can see how many rods there are as opposed to cones. So this would be at the periphery of the retina. If you were looking at the fovea centralis, it would have a ton of these cones and no rods. All right, the lens, which is right here. It is biconvex, crystal-like structure. Biconvex, the other thing that you know that is a biconvex crystal-like structure is a magnifying glass. And if you've ever been a kid who is bored, you have perhaps used a magnifying glass to start a fire. You are all home with nothing to do right now. If you are home with nothing to do, find a magnifying glass in your house. Probably have one. Go outside and light something on fire. Don't light your house on fire. Light something very small on fire and then put the fire out. That is a perfect model for how your lens should work. Light should come in. The lens, the suspensory ligament, and the ciliary body should pull the lens in a shape where the cone of light hits the fovea centralis perfectly. About that lighting things on fire. One, disclaimer, don't light things on fire. If you do it, you have to do it on a sunny day, and you're better off doing it when the sun is high in the sky. Otherwise, it requires a great deal of skill. In normal years, I give my students, we go outside with a bunch of magnifying glasses, and we light paper on fire, and that's like part of our lab, is lighting paper on fire. And so it does focus that light at the fovea centralis, and again, if it does it imperfectly, you will have some sort of vision problem, blurriness of some sort. All right, in the anterior segment, the front part of the eye, you have aqueous humor. is derived from the plasma, like your blood, and you have a little bit more or less of it, and that causes interocular pressure. You know this. They test your interocular pressure when you go to the eye doctor. They push on your, there's like a little thing that pushes on your cornea, and it sucks. Like, it's one of the most uncomfortable things ever. And when it, if you, and that tests your interocular pressure. If your interocular pressure is off for some reason, it can affect your vision, make it a little bit blurry, because again, that's just one of the things that light has to pass through to get to the fovea centralis. So because it is derived from plasma, it has glucose and oxygen in it. So it is going to provide the nutrients that the lens and the cornea need, because there's no blood vessels there. But they are in fact living cells, or living tissue. And it is a circulatory, aqueous humor is a circulatory substance. So it is going to be reabsorbed into venous blood through the scleral venous sinus, which is down here, and there's one up here. The scleral venous sinus, or one of the best names in anatomy, the canal of Schlemm, where Schlemm has two M's. I much prefer the term canal of Schlemm because it is fun and the world needs more fun things in it. However, you may also call it the scleral venous sinus, which is a more sort of modern descriptive term. So essentially, aqueous humor comes out here in the anterior cavity, and it comes back and it goes back to the anterior segment, and it comes out of the canal of Schlemm. In the back of the eyeball, the posterior part, you have vitreous humor, and vitreous humor is a gel-like substance. It is not something that circulates. You have it all for your whole life. And if you dissect an eyeball, which is something you may have done at like nature camp in 6th grade or something, it's the jelly-like stuff in the back of the eyeball. And it maintains the shape of the eye, and it is clear so light can pass through it. So you would find in here in the posterior segment, you have vitreous humor. All right, that ends it for now. I'm going to do a separate video for the eyeball lab and the vision lab. So that does it for now.